Perfect. Thanks. Well, I'm going to present our work on partial alignment of spatial transcriptomics data. This is largely work of Xinhao Liu and Ron Zeira. Xinhao couldn't be here due to visa reasons. Ron couldn't be here because he's off making money in a tech company. So you're stuck with me, the senior author, which means if you ask me hard questions, I may not be able to answer them at the end. At least someone got the joke. Thank you. Many of you know about spatial transcriptomics, which is the ability to various technologies with the ability to measure gene expression from thousands of locations at a tissue all at once with both the gene expression information and the spatial information recorded. If you didn't know about these technologies, you heard about them from Ziv's talk. There's a multitude of such technologies. The one I'm going to primarily be focused on are sequencing-based technologies. Most prominent among these is the Visium technology, which measures on an array of thousands of spots across a tissue, gene expression information, and these are in spots that are, in the current technology, about 55 microns in diameter, so it's not single-cell resolution, but still quite valuable because we get this additional 2D coordinate along with our usual high-dimensional gene expression measurement. And there's other related technologies. I put Slideseek up here, which has slightly different resolution, different coverage parameters, and even newer technologies like Stereoseek, which I'll mention at the end. So there's a number of applications of these technologies. One of the most prominent is just to think about clustering spots as we would with a gene expression measurement in the past. But now with this additional spatial information, we can look at the spatial organization of cell types. Here's an example from the prefrontal cortex where you can make out these distinct layers of the cortex with different compositions of neurons. An example in tumor where you can make out tumor versus normal, but now with a spatial context. Looking not at cell types, but at individual genes. Now with spatial transcriptomics, we can look at the spatial patterns of expression of this gene across the tissue. Now these are exciting applications, and there's others, but one of the big challenges with this data is that it's very sparse. If you're used to single-cell RNA-seq, this is similar. Depending on the technology, perhaps even more sparse than single-cell RNA-seq. And so, instead of getting this beautiful picture of this gene that's expressed in one of these layers, you instead get this very sparse information. So we and others started thinking about how you can overcome sparsity, and there's various ways you might do this. And the approach we looked at was to combine information across multiple slices, because often biologists were generating data from multiple replicate slices from the same tissue. And so we thought if we can combine this information across the slices, we could overcome perhaps some of the challenges due to sparsity, but more interestingly, we could also perhaps recover some of the 3D information from the tissue. And then you could use this combined data for all sorts of downstream applications, cell type discovery, cell-cell communication, et cetera. This figure right here is actually from It's a beautiful figure, better than my figures, because it's from News and Views that was part of our paper where we first did this alignment with this method called PASTE. This was at Recom 2 years ago and then published in the journal form last year. And so the idea here is we take multiple spatial transcriptomic slices and then we align them either in pairwise first, and then we can stack these alignments in 3D, or we integrate all the slices into a single center consensus slice to help overcome the sort of sparsity. So we're really excited about this method, and we started working on applying it to many of the more recent data sets that were out there and in collaborations on our own. And we quickly discovered some of the limitations of the algorithm that we hadn't fully appreciated. And most importantly is that in many data sets, the slices only partially overlap. And this is for a variety of reasons. One sort of just almost a technical reason that when the experimenters put the tissue slices on the array, sometimes they're sort of shifted. They're not always lined up as they were in the tissue. So you can see here an example of that from some data from a collaborator to slices from a liver metastasis. And just visually, you can see that they're just kind of shifted on the array. Perhaps more interesting biologically is, depending on how far apart these slices are in the tissue, they may have actually quite different cell composition. And so we wouldn't expect in either of these cases to be able to align them across the whole slice. So what we sort of needed to develop was an approach that would allow us to do these partial alignments. So if you're, I guess the analog from sequence alignment would be that, our existing method was sort of a global alignment, and what we need is something like a local alignment. So in this new work, in this paper this year, we presented a sequel, PASTE2, which is partial alignment of these multi-slice data sets. And so the idea here is to be able to identify the overlapping portions of these slices in some automated way, and so have some way of actually estimating the fraction of overlap, and then aligning them in pairs, stacking them up in 3D when we can. And then while we were sort of improving the algorithm, we also decided to make use of some of the information in images that's available from some of the technologies. And so we can also use the image information along with the transcriptomic information to get better alignments. So let me sort of walk you through a few of these points in the next few minutes. So first, the sort of basic idea of how we do the pairwise alignment. So the idea here is we've got from a spatial transcriptomics experiment our usual gene expression matrix, sparse. We've got now this spatial information, and so we want to align spatial transcriptomics to spatial transcriptomics, so we want a mapping from spots to spots. So we think about what properties do we want this mapping to satisfy. And so first, we would want, if we align spot I in one slice to spot J in the other, that their expression values should be similar. And so we want to preserve somehow the transcriptomic similarity. And then similarly, we want to preserve somehow the geometric information. And so there's various ways you can think of doing that. But the way we thought about doing this was thinking about pairs of points in one slice. And if we have a good alignment, then the distance between the pairs here that are aligned to pairs here should be similar. So this is a way of encoding this notion of spatial similarity. And then the third criteria is that due to various reasons, and for the partial alignment even more so, we don't expect to find a one-to-one mapping between these two slices. And even in the case where the tissue slices are very similar, there's still technical dropout, and we don't measure information at all spots. So we want not a one-to-one mapping, but a probabilistic mapping. And so the technology we use to sort of encode such a mapping is a framework from machine learning called optimal transport. And the idea here is to think of the spatial transcriptomics data as a probability distribution. So we've got this sort of distribution over these spots. And in optimal transport, you figure out a map that aligns one probability distribution to another probability distribution. Now, a common metaphor of this is, rather than think abstractly about probability distributions, people sometimes describe this as a mound of dirt. So you want to move one mound of dirt to another mound of dirt. You can do that many different ways. So the interesting part here is to try to do that in a way that minimizes the amount of work which is encoded via this transport cost. Okay, so we want to find this mapping to minimize this cost. And now in the partial setting, what we want is called partial optimal transport, which means we're not going to map all of the dirt from one mound to the other. We get to choose only part of the dirt to map. So writing this down mathematically, what we want to find here is this map pi, and we want to do this in a way that minimizes this cost. And the cost has two terms. One is the dissimilarity between the aligned spots, so this is our expression dissimilarity, and the other term is the spatial term that minimizes the distances between the aligned spots. Both these terms are weighted by this probability map pi, so it's a sum of these two weighted terms, and then there's this parameter alpha that balances these two criteria. And then the new part here is that we don't map all of the spots, we map just a fraction of this mass and this fraction S. So this is a combination of two ideas that exist already in optimal transport, and partial optimal transport and fused Gromov-Wasserstein optimal transport. So you put these together and you get partial fused Gromov-Wasserstein optimal transport. And we have a way of optimizing this function that's fairly efficient. Okay, but a key thing here is how do we choose this parameter S? This is actually a pretty tricky thing to do, and like many problems, you have this sort of model selection issue. If I give you the value S, we can actually optimize the subjective function. So to figure out the S, we came up with a somewhat reasonable heuristic. There's probably other ways to do this, but the idea here is that you look at the maps you get as you vary S when S is large. It's a very sensitive touchpad, I apologize. When S is large, you map everything, but you may not be getting the best mapping. As you decrease S, you map fewer things. And so we look at the convex hull of the aligned region. As you decrease S below the true value, this convex hull, the aligned region, actually doesn't change that much. But what does change is the boundary between mapped and unmapped spots. And what we look at is, as we decrease S, this boundary of mapped to unmapped spots start to increase. And when you get to about half of S, what tends to happen is that there's just a lot of unmapped spots here that give a high boundary. So you can sort of see this sort of coherence here below S. And then when you get to S really low, what happens is that most of the slice is unmapped, and so this boundary tends to shrink again. So we find the peak of this function, which I've written down in this edge inconsistency score, and it tends to give us a decent estimate of S. The other thing then we can do, once we have these pairwise alignments, we have this map pi, is that we can then align them to a common coordinate system by finding the rotation and translation that maps coordinates from one slice into the other. This is what's called a weighted Procrustees problem, here generalized using the weights pi that we get. And so now here, when we take these two slices and we align them in the common coordinate system, I've stacked them on top of each other, and you can kind of see the alignment. You can actually see it in part down here, this one slice being unaligned. Finally, we can add on information from the images, which is available in some technologies, in particular Visium. So here we just add another dissimilarity term, not just expression information, but we add on dissimilarity due to the RGB values in the pixels. And since we didn't want to have to solve another model selection problem, we just weight these equally with a half. But of course, you can go and weight them in a different way if you have a way of choosing that parameter. All right, so how does it work? So in order to test it, we first generated some partial alignments from true spatial transcriptomic slices. So this is data from the human dorsolateral prefrontal cortex. This was a data set where they had done spatial transcriptomics with 4 slices from 3 different patients. And so we create these partial alignments by cutting out consecutively the left or the right parts of these 4 adjacent slices. And so there's some ground truth labeling here from manual annotation from the original paper. And so then we can evaluate how well we're doing in getting these partial alignments by measuring across the slices how well do we preserve these annotations into the different layers. And we see PASTE2 here in blue with the highest value of this measure, which we call the label transfer accuracy, beating our original method PASTE and 2 other methods, Pomona and Tangram, that are made for aligning single-cell RNA-seq data sets that don't use the spatial information. So without the spatial information, not performing nearly as well. And so when we stack up slices, just showing here 4 from 1, you again see this middle region from the left and right that's aligned pretty well with partial PASTE. If we add on image information, this, depending on the images, sometimes helps dramatically. In this case, this is, again, that same dorsolateral prefrontal cortex. The images by themselves don't look actually that informative. Here again, we cut out the left and right slices to create this partial alignment. What we can see is without using the image, this one layer right here, this is layer 6, you can see in, I guess, brown there, tends to have really low sequence counts. And so it doesn't align very well when you use just the gene expression information. But when you incorporate the image, you recover actually this middle layer aligned 6, and you get this nice contiguous partial alignment as we would expect. Again, the blue are the unmapped regions due to the fact that we cut out from the left and the right parts of these 2 slices. So finally, we applied this to a number of real data sets. We're doing many more with cancers in collaboration. With several cancer researchers, but I'll show you instead a data set from a different type of technology, Stereo-seq, which Ziv also mentioned this technology. This is data from a Drosophila embryo, where they had 16 slices from this embryo. You can see that these 16 slices, although sort of adjacent in the tissue, have different sizes and slightly different tissue composition. And so then when we align them with PASTE, we can again try to assess how well we're doing in our alignments by looking at how consistent we are in mapping the cell types from one to the other across in pairwise. Again, this is using the original annotations from this paper. We can see PASTE here in blue doing pretty well, not always perfect, sometimes PASTE is better, but for the most part, PASTE2 sort of dominating because of these partial alignments. Now, interesting, the partial alignments here are in part just because of the geometry, because when you stack up all the alignments, you see, of course, that the embryo is this ellipsoid shape, right? So there's a different scale at the bottom slice. It's much smaller than the slices towards the center of the embryo. And so the PASTE2, the partial PASTE, is able to kind of recover this type of structure. All right, so in conclusion, I showed you this new approach that is really all about getting these partial alignments, which for many data sets, as we've sort of run this on many data sets, we see that the partial aspect of the alignment is really quite important due to just the geometry of the tissues, due to the different cell composition. Depending on how far apart the slices are in space, the cell type composition can change quite dramatically. A model selection procedure, sensitive mouse here, model selection procedure to actually try to get this overlap percentage, incorporating image information when available, and we hope that this will provide a useful tool for doing this sort of multi-slice integration across various spatial transcriptomics data sets and building these 3D atlases in tumors and in normal tissues. So with that, I'll conclude and acknowledge, again, first Xinhao and Ron, who were the co-authors of this paper, and other members of the group who contributed feedback throughout the process. You can find the preprint and the software online. And if there's time, I'm happy to take questions. Thank you.